The advancement of modern medicine has been a driving force accelerating innovation in the global pharmaceutical industry. Established in 2009, Biocytogen is a biopharmaceutical company that also provides revenue-generating preclinical research services. Over the past decade, we have developed innovative gene editing technologies and antibody drug development platforms, which serve as the cornerstone of our continuing growth. We intend to develop and commercialize antibody-based drugs that improve human health. As of the latest practicable date, we have built a product pipeline containing 12 drug candidates. Our core product, YH003, a recombinant humanized agonistic anti-CD40 IgG2 monoclonal antibody, showed a favorable safety and efficacy profile without liver toxicity in a Phase 1 clinical trial. YH003 is being evaluated in combination with anti-PD-1 monoclonal antibody as a first-line and second-line therapy for patients with pancreatic ductal adenocarcinoma. Another core product, YH001, a recombinant humanized anti-CTLA-4 IgG1 monoclonal antibody, also showed a favorable safety and efficacy profile in Phase 1 trials. Honing our technological capabilities, over the past several years, we established advanced gene editing platforms, animal model platforms, antibody discovery platforms, pharmacological and pharmacodynamic service platforms, and a strong clinical development team. Together, these platforms streamline the entire drug development process to fuel the continuous production of novel antibody drugs. Leveraging our proprietary gene editing technologies, we have developed a series of target humanized mice and a series of severely immunodeficient mice. We have established an international high-standard animal center with full ALAC accreditation that houses an extensive portfolio of animal models for large-scale animal production and in vivo efficacy studies. With these models, we have built competitive pharmacological and pharmacodynamic services. Our in-house drug pipeline and external collaborations have endorsed our unique in vivo antibody drug screening platforms. With our chromosome engineering technology, abbreviated SUPCE, we have developed fully human antibody platform RenMice, consisting of 2 fully human transgenic mice lines, namely RenMab and RenLite. RenMab mice contain the full human variable region, and RenLite mice contain the full human heavy chain with the common light chain. RenMice platforms can generate a highly diverse human antibody library for further development of antibody-drug conjugates, ADC, bispecific antibodies, and bispecific ADCs. To address difficulties in developing antibodies for new targets, we launched Project Integrum, an innovative large-scale antibody drug discovery program. We intend to knock out each of the over 1,000 potential antibody drug targets one by one in our RenMice models. We will also use our evidence-based in vivo drug efficacy screening platforms to obtain antibody drug candidates within the next 3 to 5 years. Since the launch of Project Integrum in March 2020, we have knocked out more than 980 target genes and carried out antibody discovery and screening. Project Integrum allows us to circumvent traditional methods to identify preclinical candidate compound, PCC antibodies, for potential targets, which significantly accelerates the discovery and development process of novel therapeutic antibody drug candidates. Over the past decade, our technologies and products have brought us extensive recognition from a number of leading partners in the field. We have collaborated with 9 of the top 10 largest pharmaceutical companies globally, reached 24 drug co-development deals with 15 partners around the world, and licensed our RenMice platform to 14 pharmaceutical and biotech companies. These continued collaborations are expected to generate considerable economic return to us and accelerate the development of many potential antibody candidates. Our seasoned and visionary management team possesses solid scientific and business experience that has successfully driven the global expansion of our business. In the future, we will continue to advance Project Integrum and strive to become a global source of new drugs. We aim to accelerate the commercialization of our pipeline products and make every effort to implement our global strategy through more open collaboration. Our goal is to become a world-leading biopharmaceutical company that contributes to the improvement of human health.